Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Moving AAV Gene Therapy Beyond Rare Disease to Address Large Patient Populations with Chronic Disease. I'm Jokubas Likauskas, an editor at BioInsights, and joining me today is Dan Oliver and Jackie Lee, who will explore how innovative pipelines, clinical strategies, and novel technologies like TESSA are making AAV gene therapies commercially viable for cardiometabolic, age-related, and animal health conditions. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we will try to get to them during the session. Now, I would like to introduce our presenters. Daniel Oliver, CEO and co-founder of Rejuvenate Bio, leads the development of novel gene therapies for age-related diseases, building on his entrepreneurial background and engineering expertise from Caltech and Harvard. Ji Hyun Jackie Lee, a process development scientist at Advanced Therapies, specializes in viral vector purification, focusing on AAV and LVV with a biochemistry degree from Temple University. So without further ado, I'll hand over to Dan to kick off today's presentation.
Hey, thank you so much. I'm excited to talk to you all today, excited to answer questions at the end of the presentation, but I'll just jump right into it. We certainly have seen a lot of critiques of gene therapy over the last, let's call it 18 to 24 months. A number of those critiques have been that it's too expensive, only relevant for niche applications, and that there's been severe manufacturing struggles with it. And so here's some headlines, and obviously these are representative of a number of different things people have critiqued gene therapy on today. Our goal really is to show you that these are not inherent problems with gene therapy. They are issues that are very much solvable, both with new technologies, but also with better choices for how gene therapies are developed. And so I'm excited to be working with the WuXi team to show you how we are actually taking gene therapy not only into very prevalent patient populations on the human side, but also have driven the manufacturing costs of these therapies such to the point that they're relevant for treating indications in animal health, so treating people's pets as well. So just as we move forward today, we'll be covering three major areas. First is the marrying of the delivery targets and manufacturing yields to create scalable gene therapy solutions. And so I'll be starting about talking how it's not just about your manufacturing cost, although it's an incredibly important part, but there's a huge amount of decisions that need to be made literally from day one to make sure you're creating a scalable gene therapy. Secondly, I'll be moving on to how we're validating this platform in the animal health market. So before we ever go commercial in human health, we will have loads and loads of pets that have taken the therapy and are treating very, unfortunately, severe issues in animal health, things like mitral valve disease. And then finally, look forward to hearing from Jackie around Advanced Therapies system, TESSA, which is a huge component of making this all possible for scalable gene therapy. So just a bit about Rejuvenate Bio before I fully dive in. I'd be remiss not to mention some of the folks that helped start this and run the company. This includes my co-founder, Noah Davidson, as well as Professor George Church. Noah was a postdoc in George's lab and invented this technology. But one of the most rewarding parts of being able to work at Rejuvenate has been the expansion on bringing really amazing people onto the team. This includes Deborah Asheim, who's our Chief Medical Officer, formerly at Stride Bio, and a number of other gene therapy roles, and really is basically the leading medical expert on cardiac gene therapy. And so it's amazing to be able to work with Deborah. Second, we've also been able to pull luminaries in basically each key portion of the development we're doing for our gene therapy. So this includes Dr. Catherine Hai of Spark Therapeutics fame, Frank Greenway, or I should say Professor Frank Greenway from the Pennington Biomedical Research Center at LSU, who helps us with our metabolic work. And then also Hugh Calkins, who is the leading clinician for arrhythmogenic cardiomyopathy. And then finally, on the animal health side, Fabian Couch, who was the former head of Boehringer Ingelheim Animal Health R&D. Awesome. So this jumping into our first section, I'll be talking about how our liver-directed approach with secreted proteins allows us to have a scalable solution. I'll talk about our lead product, gene therapy encoding for FGF21, a longevity gene with multiple applications. Talking about where we initially focus this technology, and then finally talk about the best-in-class positive pre-IND, best-in-class data and positive pre-IND feedback that we've been able to generate with our first gene therapy. And so first off, there's a huge demand for cardiometabolic treatments, and they really are driving unprecedented sales and valuations in the pharma industry. And so I'm sure all of you are aware of Eli Lilly and Novo's products, the GLP-1 class, and I certainly don't want to just make a one-to-one comparison what we're doing, but what I think is truly intriguing about these products from our perspective is this idea that treating chronic disease in multiple chronic diseases is not only possible, but is hugely commercially viable and is also very, very beneficial for patients. And so the demand for these products, not just because they cause weight loss, but because they treat things like heart failure, metabolic disease, renal failure. I've even seen data suggestive that they're helpful with things like addiction, is really a great illustration that there's a class of therapeutics that are out there that we can build that have a laundry list of positive benefits across multiple applications versus that typical laundry list of negative side effects that you see with therapeutics. And so we are super excited to build a technology platform that allows us to augment or replace GLP-1s moving forward. And so we utilize safe gene targets with extensive human and large animal data. Our delivery approach provides well-tolerated, durable expression that addresses issues with protein therapeutics. And our delivery via the liver allows us to achieve systemic effects ideal for chronic disease. And so marrying these all together really allows us, as Kathy High says, to push gene therapy into this realm of complex chronic disease and not be pigeonholed into monogenic gene replacement. Awesome. So our first gene therapy utilizes FGF21 as the payload. And it's really the ideal lead program to treat chronic disease in humans from a gene therapy standpoint. FGF21 has been proven to be well tolerated with durable expression with one-time administration. There's been limited safety issues in late-stage human clinical trials with synthetic protein analogs of FGF21. And we have also generated over 3 years of large animal data showing sustained expression, no acute reaction to the gene therapy, or long-term negative side effect from overexpression of our key proteins. FGF21 activates key pathways associated with numerous prevalent chronic diseases. And so it metabolizes excess fat, increases insulin sensitivity, and decreases triglyceridemia. It decreases inflammation, fibrosis, and arrhythmias in cardiac disease. You'll see why that's so important coming up. And it reactivates and strengthens the immune system. And then finally, our liver-directed approach is the ideal modality to deliver FGF21. FGF21 is naturally created and secreted from the liver. So by delivering it via gene therapy targeting the liver, we avoid compliance issues and eliminate side effects of other therapeutic classes. So here's some of the data we've been able to generate to show just how powerful delivering a gene therapy encoding for FGF21 can be. So this is a paper that launched us out of the Church Lab and really demonstrates the broad applicability of being able to deliver these types of factors. And so in 3 separate experiments, we were able to demonstrate that an upregulation of FGF21 delivered via a AAV gene therapy primarily targeted to the liver was able to treat a model of heart failure. This is ascending aortic constriction, was able to deal with obesity. We actually were able to fully reverse type 2 diabetes phenotype as well, and also basically drive mice back to their typical body weight with no loss in muscle mass or change in bone density, even when put on a high-fat diet. And then finally, also were able to show robust effects in a model of renal failure or kidney failure. And so the point here is to really create therapeutics that have broad applicability such that you are able to scale through different patient populations to get these types of benefits out there. And you can really see that through our proposed pipeline. So I'll start with the cardiac version, but we'll also dive into the metabolic side as well. And so with this kind of broad applicability, as you can imagine, there's multiple different areas, not only you can go to, but also where you could start. And so in a second here, I'll be talking about why we selected desmoplakin arrhythmogenic cardiomyopathy as our lead indication. But I think the really key takeaway here is the ability to expand out with these same drug products, same manufacturing process, same drug product, into not only multiple variants of arrhythmogenic cardiomyopathy, but into very broad cardiac diseases, as well as moving into very large metabolic diseases moving forward. And this just gives a very different profile for a gene therapy, whereas people have in the past, in some sense, have to be pigeonholed in very specific monogenic diseases. And maybe there's a delivery platform that they can continue to utilize across multiple indications. But this is literally the same drug product that can scale across all of these different applications. And so we decided to start with desmoplakin arrhythmogenic cardiomyopathy. It's a clearly defined patient subset ideal for our first treatment, RJBO402. And so delivering FGF21 to these patients addresses very key aspects of the multiple phases of their more fibrotic desmoplakin variant of arrhythmogenic cardiomyopathy. So these patients, just for your edification, have a mutation in one of their desmosomal proteins. These are the interconnective chains between their cardiac cells. And when one of those chain links breaks, a couple of key things happen. They see an uptick in arrhythmias. This arrhythmias drives inflammation and remodeling and fibrosis. And obviously having a break in one of those chains decreases the structural support of their heart. And so FGF21 actually deals with all of these issues, even though they aren't even though it isn't a direct replacement for the malfunctioning desmoplakin gene or other mutated desmosomal protein. We are utilizing this more generalized approach to tackle the desmoplakin variant, which is not addressable with standard gene replacement. The desmoplakin gene is too large to be packaged into an AAV, and so direct monogenic gene replacement is not appropriate there. There's a robust initial patient population with immediate expansion opportunities here. And so there's approximately 30 to 50,000 DSP ACM patients in the US. But again, since we're taking this more generalized gene agnostic or genetic agnostic approach, all variants or ACM should be available to us to expand into, which is something on the order of 100,000 patients. And that allows us to drive into these large cardiometabolic conditions as well. One other thing I'll mention about ACM patients is they have both, fibrotic and arrhythmic involvement in their heart failure. And so it opens the door to, multiple different types of follow-on cardiac applications. And then they also are one of the few patient populations who their doctor tells them not to exercise. Exercise has been shown to stress their hearts and can exacerbate their condition. And so these patients, as is prevalent in the general population, also tend to have metabolic conditions. And so all of these patients can provide insights through secondary endpoints into how this type of gene therapy not only would be tolerated, but also into efficacy readouts in these patients suggestive of being able to move into these more prevalent cardiometabolic conditions. But saying that, I think it's really important to stress that we are highly focused on being superior and a superior treatment to anything on the market for the treatment of DSP ACM. This is a particular mouse model that we've run. In this mouse model, they knock out one of the desmosomal proteins. In this case, it's the PKP2 protein. As you can imagine, when you do that, you see an increase in arrhythmias in these animal hearts. These are mice, arrhythmias or ectopic beats. And so by giving our gene therapy, what we've been able to demonstrate is a severe reduction in those arrhythmias or ectopic beats in these animals' hearts. If you compare and contrast our approach to folks that have done monogenic gene replacement with the PKP2 gene, these folks typically have done, AAVs that are tropic to cardiac or muscle cells. delivering a functional copy of the PKT2 gene, what you see is actually superior reduction of arrhythmias using our approach at far lower doses. And so at a dose that's about an order of magnitude less than monogenic gene replacement does. Maybe asking yourself, how is this possible? I mean, this is a monogenic, this is a disease driven by a monogenic mutation. Certainly a monogenic gene replacement approach would be the superior, superior solution. Well, the answer is, the devil is in the details here. To have these approaches work, you need to get your beneficial gene/protein to the cells who need it. For us, we utilize the power of the fact that FGF21 is a secreted protein. So even though we aren't infecting very many cardiomyocytes and we aren't actually expressing FGF21 from really any of them, the fact that FGF21 is then secreted in the bloodstream allows us to get into basically every cardiomyocyte with their FGF21. And so even though these folks are utilizing a cardiac-directed approach, they have to basically inject their patients with high doses of gene therapy to try to get that virus into every single cardiomyocyte. Because if they don't get it in with the virus, then it's really not going to have benefit from this. And so I think this goes back to this idea that how you build your gene therapy, both your delivery, the gene selection, is incredibly important for the scalability of the gene therapy. And so again, it's not just a question of moving into cardiac disease, but we also have applications across the metabolic space as well. And so we think an interesting opportunity to enroll patients and go after patients that aren't responders to GLP-1. Also possibility to utilize it as a combination treatment with GLP-1. And then also there's been robust data showing effects of FGF21 in MASH patients as well. And so some of the data we've been able to generate in the metabolic space is that a single administration of our therapy reverses a weight gain in type 2 diabetes phenotype. And so mice put on a high-fat diet, were treated with our gene therapy. Over the course of approximately 40 days, they returned to their normal body weight, even though they were continued on that high-fat diet. During that time, the treated mice saw no change in bone density and no loss in muscle mass. To compare and contrast that to GLP-1s, obviously, who are seeing really robust weight loss, but also have the negative side effect of pretty drastic reduction in muscle mass as well. We also saw reversing of a type 2 diabetes phenotype as well. So mice on this basically saw a decrease in their HOMO-IR score, which looks at insulin resistance after receiving the treatment. We've also seen in the literature, I should say, that unmodified FGF21 protein can dramatically reduce weight in non-human primates. And so we have seen, GLP-1 type weight loss from a series of FGF21, unmodified FGF21 injections. This is important to note because as folks have modified the protein to make it viable from a half-life perspective, it seems to have modified its ability to reduce weight in large animals. And so I guess the point I would be making here is this way of delivering FGF21 via gene therapy, so you're able to counteract the half-life issues of FGF21 by still delivering unmodified FGF21 via the gene therapy versus a stabilization of the protein that seems to affect its dynamics in the body, is a superior way of treating these types of indications. So gene therapy delivered here with unmodified FGF21, it really offers superior weight loss to modified FGF21, to my point in the last slide. And so there is a difference between a protein therapeutic that has to look at half-life issues versus our gene therapy, which is able to basically give natural FGF21. And we can actually see superior weight loss even to GLP-1s in different contexts. It's not just about weight loss, though. These patients have also demonstrated a number of different metabolic metrics that have gone in a positive direction based upon delivery of these therapeutics. And then finally, we also seem to eliminate a number of the class effects associated with protein therapeutics, and really is not really an appetite suppressant. So it's helping to curb muscle loss so that the weight loss you're seeing is truly what people want, which is not necessarily just weight loss, but in some sense becoming leaner. You want your weight loss focused on a reduction of excess fat, not of a loss of functional tissues such as muscle tissue. Awesome. So now I'd like to move into talking about the use of our gene therapy for the treatment of major indications in animal health. And so we've been able to generate key positive data in multiple blockbuster indications on the animal health side, validating our approach. We've actually been able to sign 6 commercial partnerships with leading animal health companies, strategically de-risking this and showing that there is appetite for these types of therapies. And then we actually have 3 additional partnerships in negotiation that we hope to move forward this year. And so just a little flavor of that, we've had strategic buy-in of leading animal health players that validates our approach in humans. And so first example is a commercialization partnership with a leading animal health company called Phibro Animal Health. Phibro will lead all marketing, manufacturing, distribution, and sales efforts for our first therapy in dogs called RJBO1C. Similar to our therapy, we're moving forward into arrhythmogenic cardiomyopathy in humans, but incorporates another gene called STGF-beta-R2. And it's treating the leading indication in the cardiac space for dogs called mitral valve disease, which I'll get into in a sec. We receive a large royalty payment in exchange for this. The second, we've development partnership with a leading animal health company. This is one of the biggest animal health companies on the planet. This animal health player receives worldwide rights to our second therapy, which is a treatment for osteoarthritis. They're actually paying for all the development and commercialization costs for this therapy. We receive a robust set of pre-commercial milestones and commercial milestones for moving this forward. And so again, huge amount of validation here that we've been able to sign these partnerships, and then we have more likely moving forward in 2025. So just to talk a little bit about the specific applications we're going into in the animal health space, I want you folks to get kind of a flavor for how many dogs really have these issues and how big of an issue it is both for the dogs and their pet owners or pet parents. And so mitral valve disease is a huge unsolved challenge in animal health. So it's a death sentence with current treatments. It's caused by a malfunction of the mitral valve, which unfortunately eventually leads to congestive heart failure. And so if you folks remember your anatomy, there's 4 chambers to the heart. The mitral valve basically stops backward flow into the left atrium from the left ventricle. And so if you have a malfunctioning mitral valve, what you get is blood flowing backwards through the heart that hits the back wall of the left atrium, causes inflammation and fibrosis, expands the left atrium, and then unfortunately drives these animals into congestive heart failure. About 7% of all dogs are afflicted with mitral valve disease, and there are particular breeds, including the Cavalier King Charles Spaniel, that have incidences approaching 80%. I think this is important to note because if you are a lover of this particular breed, or frankly of small dogs in general, it's highly likely that you will run into one of your animals unfortunately developing mitral valve disease. And so the pet parents who have experienced this with their animals, unfortunately, oftentimes have experienced it multiple times. And so there is a really robust community around looking for solutions to this. In some certain akin to a patient advocacy group, but obviously a pet owner group that's looking for solutions to a problem that's afflicted, pets that they view as basically part of their family. Represents a blockbuster business opportunity. Mitral valve disease is also easily diagnosable. So this is key in animal health. Obviously, dogs can't communicate verbally with us. And so being able to pick up on issues with them is very key to being able to treat them. And so MVD is diagnosable with a stethoscope. It initially presents as a heart murmur, and then the dogs go up for a full cardiac workup, particularly with echocardiograms. There is an existing standard of care medication called Vetmedin. Is a fairly robust seller, several $100 million in sales annually, but unfortunately does not treat the underlying disease, but lowers the load on the heart. It's an inodilator. So moving forward, what have we been able to prove in this space? Well, we've been able to take dogs on mitral valve disease and basically delay the progression of their disease by approximately 2 years. And so, as you can imagine here, when the colloquial thing is 7 human years for a dog year. Similarly, their heart failure progresses quicker than our heart failure would in this same type of context. And so delaying out 2 years for dogs is gigantic. And so dogs given our gene therapy basically have seen lack of progression to cardiac death or movement into full congestive heart failure. We've also been able to demonstrate an increase in key heart metrics as well. So we see an increase in heart function in these dogs given our gene therapy, as well as a decrease in heart damage measured by left atrium size as well. And so really exciting data. We're excited to push this forward with our partners at Wuxi to allow this to continue to move forward to actually help these dogs out and treat this, unfortunately, life-threatening issue with them. And so to follow up there, just to give you folks all a perspective, we are, this is a large market opportunity in animal health. And so, I think people rightfully think of animal health as typically smaller market sizes than human health, but there is a robust opportunity in mitral valve disease. So as I mentioned, there's a current treatment that's generating multiple hundreds of millions of dollars per year with this treatment. And then there are a huge amount of dogs that go untreated here. And so as you can see here, there's a willingness to pay multiples of thousands of dollars to treat life-threatening indications in animal health. So a couple of examples here that you can probably pick out. And frankly, if you've had a pet, you've probably unfortunately experienced the high vet bill. But anytime you have an animal have any sort of surgery, it's at least $1,000 to $2,000 just to put them under anesthesia. And that doesn't even deal with the particular intervention you're looking to have. And so, large, robust set of folks that are paying, thousands of dollars to have surgical interventions for their animals. There also is a really robust market for the treatment of cancer in dogs. And so chemotherapy and radiation treatments can range from $5,000 to $10,000. And it's also important to note there's demonstrated market data that there are hundreds of thousands of dogs that are administered therapies that are in this kind of price range moving forward. And so to compare and contrast that to the cardiac space, 2 years of being on vetmenin would cost a pet parent something like $2,500. It's not a particularly cheap medication, and it's also difficult to administer. You're taking a relatively small dog. These dogs are typically around 10 to 15 pounds, and you have to basically give them a pill at least once a day, often twice a day, by shoving it down their throat, which is not a pleasant thing to be able to do. And so one-time intravenous injection of a gene therapy, I think, will look quite attractive to these pet parents. We've actually done this in the context of the data I showed you on one of the previous slides. But the appointment for actually administering this therapy is less than a half hour. We basically, hook up an IV to the hind leg of the dog, then administer the gene therapy, allow it to perfuse in over the course of something like 10 to 15 minutes, then watch the dog for another 10 minutes, and then it goes home. And then it's one and done. And so, look, if you think about how many dogs have mitral valve disease, there really is a robust market opportunity here. And so 5% of the 1.5 million dogs that are diagnosed with the stage of MVD that we would intervene at would yield something like 70,000 annual sales that would be greater than $200 million in revenue. And so I think gene therapy is a very, very viable solution for these types of issues in animal health. And by choosing non-specific targets and choosing delivery paths that allow us to minimize those but still have systemic effects, we're able to create gene therapies that are highly relevant to treating these very prevalent chronic indications, not only in the animal health space, but also in the human health space. So I'm excited here to be working with the Wuxi team that allows us to enable to have a spread, even at these types of price points we're talking about in animal health. And I'm excited to hand it over to Jackie now to allow us to move forward and hear more about the Tessa manufacturing platform. Jackie, thanks for joining me today.
Yep, thank you so much for the introduction, Dan. As I said, I'm Jackie here at Advanced Therapies, and today I'll be expanding on the power of the TESA technology, which is a novel solution for pioneering AAV manufacturing for commercialization, and to be especially powerful in enabling treatments for chronic and rare diseases like the work that Rejuvenate Bio does. So currently on the market, there are two possible options that we could go for AAV manufacturing. The first of which is the helper-free triple plasmid transfection platform, which is the most prominent and most well-known. This process involves using three different plasmids, which encodes for your RepCap genes, helper functions, as well as your chosen GOI to transfect the cells to produce your AAV product. While this method is attractive in that it does not involve any adenovirus in the processing and can be robust and adaptable, it does face some issues in that it is not as easily scalable the larger the scale you go due to the transfection limitations, which also involves high cost of goods and may be low yielding, especially dependent on the serotype that you choose. A secondary option you could go with is the producer cell line. So in this option, you have the cells that are already integrated with the rep, cap, and the GOI sequences, and then is initiating the AAV production using an either wild-type adenovirus or a hybrid virus. Compared to the triple plasmid platform, this method is more easily scalable with lower cost of goods and may give you higher yield, but it does involve the adenovirus inclusion, which means that you would have a downstream processing step that tries to inactivate the virus and remove it from the AAV drug product, and may not be as robust and adaptable since the cell line itself needs extensive characterization. What we like to offer from Advanced Therapies is the TESA technology, which involves the pros of both of these platforms and combines it into one easy applicable solution for the AAV manufacturing. So how does this actually work? So in nature, we see that the adenovirus generates higher yields and more infectious AAV products. But how do we actually prevent it from contaminating your final AAV prep batches? So the TESA stands for tetracycline-enabled self-silencing adenovirus. So in the adenovirus, we have engineered it so that in the major late promoter region, we have included the TET repressor binding site. And then also the TET repressor itself is downstream of it within its transcriptional control. This means that in the presence of doxycycline, it will go ahead with your normal virus replication, but in the absence, it will only go with the early gene expression and terminate the late phase life cycle, so that it will only go forward with the genome amplification. And this is regulated via a negative feedback loop. What this means is that we can go ahead with a replication incompetent TESA virus that is preventing any contamination issues within your final drug product in the manufacturing space. Now, using the TESA vectors, we like to offer two different manufacturing platforms, the TESA Duo and the TESA Pro. In the TESA Duo system, we have a already made adenovirus that contains the rep and the necessary cap genes, along with another engineered adenovirus containing your GOI. So these two adenoviruses are used to co-infect the HEK293 cells to produce the AAV GOI product. This method is more suitable for serotypes with a lower infectivity for the chosen cell line and is recommended more for indications requiring large doses or have larger patient populations. Another option is the TESA Pro, in which that it is more familiar to how AAV is made in nature using the adenovirus, and that is more of the off-the-shelf unit. So with this system, we have the already made adenovirus with the RepCap genes, and we have a pre-existing AAV GUI product often supplied by the client themselves, and use them both to infect the cells to produce the AAV. So in this case, you can achieve up to 3,000-fold amplification of the input AAV and may be more suitable for GUIs that are toxic to adenovirus or are for products that are more stringent on the timeline. For more information behind the TESA technology and in-depth analysis, I'd like to point you to an article published in Nature Communications by Wei-Hung Su and our colleagues at Oxgene, who are the true inventors behind the technology itself. Now, from the prospective user standpoint, what does the manufacturing timeline actually look like? So for either platforms, we first start off with a material transfer in which that we would receive the GOI plasmid sequence information as well as any AAV product that we could use as reference. So then most of the clients that we receive here at Advanced Therapies are already familiar with the triple plasmid transfection platform, so we would do a comparison study between that and the TESA. With the TESA Duo, we need to first start off with generating R&D seed stock for the adenovirus containing the GOI itself, and then move forward with the comparison study. From the results of the comparison, we would then make sure that our both upstream and downstream processes are sound and easily applicable for your GOI product generation before we move on to the master virus bank generation. Once the virus bank generation is complete, we will use it to infect the HEK293 cells to generate your final AAV-GOI. And at this stage, we would make sure from a process development standpoint that the process itself is fully optimized, and then any analytics that are necessary to define your product are ready to go before you enter into the GMP production phase. Now let's take a more deeper look into the actual AAV manufacturing itself. So I like to show here the upstream and the downstream processes of what we use as a standard platform at Advanced Therapies. So we have a fully defined, very robust manufacturing platform that can boost both starting and overall yields with data to support that I will show in the later slides. So first, we will start with a seed train with our proprietary HEK293 suspension cell line, which would then be infected with either the Duo or the Pro system at an optimized MY ratio. The produced AAV will be then harvested and transferred for purification, which starts off with clarification, and then through affinity chromatography for the AAV capsid capture, the ultracentrifugation or IEX chromatography for the full capsid separation, and finally buffer exchange into your chosen formulation using UFDF. So as you see, all the unit operations that are shown here are commonly used for the triple plasmid transfection platform as well. So you have a familiar framework to work with when finalizing any GMP preparation and testing details. Now, how does this actually work in application? So I would like to show here a data set that we have accrued using an AAV6 eGFP production at a 50-liter scale. On the left-hand side, you see a figure with showing the upstream harvest yield comparison. The left bar is showing the yield from the triple plasmid platform at a shake flask scale, which we're able to produce 2.1813 GC per liter of harvest. Now applying the TESA Duo system and going up to 50-liter bioreactor scale, we were able to increase the titer up to 33-fold, reaching 6.9E14 GC per liter of harvest. And then we applied this using an initial downstream process, which used UC and IX for the full capsid separation. With this robust process, we were able to achieve up to 2.7E14 GC liter as your total yield for the drug substance. And using AUC, we were able to determine that we were able to see 94% full for the purity for the final product. From there, we decided that we could optimize the process even further. So for the downstream process, we decided to eliminate the UC step and only focus on the IEX for the full capsid separation. This process allowed us to increase the total yield almost 2x to up to 5.9814 GC per liter of harvest, and still maintaining a full capsid percentage of 88% as determined by AUC. We see that the drug product has 99% purity based on SDS page and retains a high infectious titer of 1.8% of the capsid titer, showing that we were able to produce a very high quality drug product using the upstream and the downstream process. Now looking at the actual adenovirus level within the drug product, we have three major assays that we use here at Advanced Therapies. The first of which is the residual infectious adenovirus. Through this assay, we were able to see that the adenovirus level was below the limit of detection, so it gave us a negative readout. And the residual hexon DNA was at very low level of 0.009% of the AAV ddPCR titer, as well as the residual hexon protein was able to also similarly reach low levels at less than 3.9 E4 particles per mil, showing that we didn't have any adenovirus contamination within the final product. Now we would like to show you data from how it scales up to 200-liter scale, especially using a serotype that is shown to have lower yield using AAV2 and still maintaining GFP as the GOI. So on the left-hand side, you still see the upstream scale-up comparison, where we compared first the triple plasmid platform and the Tesla dual system at different production levels. The triple plasmid showed the titer of 1.4813 GC per liter of harvest, and while still at a shake fast level, the Tesla dual system shows a significant improvement at 1.1814 GC per liter. At the 50-liter scale scaling up, we see a 4.9814 GC per liter, and similarly maintaining a consistent production at 200-liter scale bioreactor at 3.8814 GC per liter, which equates to a 27-fold increase compared to your triple plasmid. And then using the optimized downstream process, we were able to still see a very robust and consistent production of a high-quality drug product at different scales at 25-liter and 2 different 50 liters respectively. So we see that we were able to see 1.2 E14, 1.5 E14, and 1.2 E14 GC per liter respectively, showing that we still maintain that robustness going from all the way from upstream to your downstream process. Now, how does this actually apply using GOI? So here I would like to show you the data that we have using the Tesla Pro system with an AAV8 GOI application at large scale at the 50-liter scale. On the left-hand side, we see the upstream scale comparison, again comparing the triple plasmid transfection platform to the Tesla Pro system. So we first did a same scale comparison at a 2-liter bioreactor, in which that triple plasmid showed us 1.6 E14 GC per liter, but then the Tesla Pro still showed us a significant improvement at 1.1 E15 GC per liter of harvest. And scaling up to 50-liter production, we still see a 5-fold increase at 7.4 E14 GC per liter harvest. And with this particular GOI and the optimized downstream process, the triple plasmid transfection was projected to see a 6.4 E413 GC per liter for the drug product. But then with the Tesla Pro, we see a 3.3 E14, a markedly higher jump at a 5-fold increase for the final drug product for the Tesla Pro system, showing that we still see a very robust and scalable process, even with the GOI application itself. So in summary, the Tesla Duo and the Tesla Pro system offer a very competitive AAV platform that can first easily increase the starting and overall yield for the upstream and the downstream processes, can easily scale up to 200 liters and beyond for your clinical and commercialization needs, be applied with a robust, well-defined production process, especially with our own HEK 293 suspension cell line, while maintaining a very high product quality profile, and therefore lowering the overall cost of goods and ultimately provide an efficient solution for AAV-based gene therapies. Thank you for your time listening to both Dan and my presentations on Rejuvenate Bio's work and the Tesla technology. I believe we are ready to take any questions from the audience now.
Yes, absolutely. Well, first of all, thank you, Dan and Jackie, for such a great and insightful presentation. And now we're going to start our Q&A. We have quite a lot of questions, but please keep them coming in, and we'll try to get to as many as we can today. And the rest we'll answer via email. So the first one is, what kind of filters do you use during the harvest? Sartorius offers a maximum liter size filter unit for humans.
Right, so I believe that is a question from me. So for clarification, we do use a depth filtration system. Although I can't specify which vendor that we use for the filters itself, we do specify different primary and secondary filters with their own retention rates specified for the AAV so that we have a clear reduction in turbidity going from the crude cell harvest slices to the clarified to move forward into the purification process. Oh,
Thank you, Jackie, for your clarification. We actually have another question. So, how long is the DNA construct used for FGF21, and are there more than one construct? And what's the percentage of full capsids during fermentation of the AAV? Would any of you be willing to answer this one, please?
Yeah, I can take a shot, and Jackie, you can correct me if I'm wrong. I don't know if we can share everything about our manufacturing process here, but I'm guessing they're asking how long we're not using plasmid DNA, and so you're using adenoviral that carries our GOI. I mean, you're making a virus bank for that, and so it's more of a question of how many runs you do with that particular virus bank would be my answer. And what I'll say as far as the empty full ratio, and like, look, we're still in the process of getting this scaled up to GMP, so I don't have a percentage for you today, but we're seeing superior empty fulls than we saw from a triple transfaction process that went through a huge amount of process development. And so we are very high amount of folds in our process. I don't know, Jackie, if you have anything to add to that.
yeah, so to support Dan's reply, although it varies depending on your GOI and how much you have optimized the GOI sequence as well as any other option parameters as well. Typically, we do see a very competitive full and empty capsid ratio compared to the original triple plasmid platform. And combined with the higher yield that we see with the Tesla platform itself, regardless whether you use the Pro or the Duo, we see that overall you'll see a much greater overall starting yield going into the rest of your downstream processing.
Okay, thank you both, Dan and Jackie. That was really insightful. And now let's get to the next question. So, what kind of QC test do you perform before the product is released to the veterinarian for injection/infusion?
So just a kind of a larger point here, and then Jackie, I think, could go into the specifics. Um, the animal health the animal health industry is regulated by the FDA. There's a Center for Veterinary Medicine at the FDA that looks at your applications for this, but really the regulatory process is very much akin. And when it comes to CMC requirements, they actually are effectively just flowing through the CBER requirements onto the animal health portion. And so there are some differences that I think are notable on the efficacy and safety end of the equation. But for the manufacturing requirements, we're effectively scaling up a full GMP compliant human process, but utilizing a GOI and obviously a therapy that's being pushed towards animal health. Just a couple of things here because I didn't cover my presentation. On the regulatory side for animal health, I think the key difference there is you don't typically go through 3 different phases of clinical trials for animal health. The typical animal health regulatory process is you file an INAD, you then get sign off on what they call your target animal safety, which would be akin to a phase 1 trial in humans. But actually, for you folks, it would look a lot like a GLP toxicology experiment. So typically done in lab animals. Depending on the modality, often is multiple doses. For gene therapy, what we're seeing is typically will be 1 dose at multiple time points. And then secondly, then you would have a pivotal efficacy trial that would be akin to a phase 3 trial in humans. In animal health, things like conditional approval are far more usual. And so Rejuvenate Bio is actually pursuing a conditional approval, which would allow us to basically begin marketing our product as we're running our pivotal efficacy trial on the back of what they call reasonable expectation of effectiveness, which we anticipate having generated from a study at Tufts with people's pets as well. But Jackie, feel free to answer particular purification questions as well. Sorry, QC test questions.
Yeah, so although I can't fully speak from a QC standpoint, I would like to add that at least here, as Dan mentioned, we treat the product, even if it's a veterinary product, we treat it as if it will be used for human use within the actual testing panel itself. So for example, to characterize the either upstream or the downstream processes within the testing, we make sure that any of our testing that we typically use for any, for example, human use triple plasmid platform is also carried over whether you use for the tested tool or the pro system as well. So for example, at least from speaking from a downstream perspective, uh, if you use a certain type of chromatography or UC for the full capsid empty ratio, as I showed in the presentation itself, you would use AUC for the full to show the full percentage. You would use different residual assays to make sure that any of the leftover particles that you may expect from for example, the downstream step operations are fully characterized within your AAV product at the end.
Fantastic. These are great answers. Thank you so much, both. I'm sure our audience appreciates it. And the next one is actually for Jackie. So, what is the most time-consuming part of using the TESSA program?
I would say for either the Tesla Duo or the Tesla Pro, the most time-consuming part would be optimizing first the MY ratios that you will use to inflect the cells to generate your AAV. But particularly for the Tesla Duo system, because there is the need to first generate the R&D seed bank to use for that first comparison study, and then to ensure that when you are going to your massive virus bank generation, the adenovirus containing your GOI sequence is still fully scalable for your virus bank generation. We need to ensure that from a PD perspective, that all the process parameters are set and settled while achieving a high yield for the eventual AAV infection. So everything is fully characterized before you go into any GMP processing. So I would say the time that it takes to make sure that all those parameters are set and all the analytics that are associated with it are set before you start off with the actual AV production is the most time consuming, and also requires a lot of details to be reviewed and carefully monitored from the client's perspective.
That's great. Thank you. Thank you, Jackie. And before I get to the next question, can I please remind our audience to keep the questions coming in, and we'll answer as many as we can now, and the rest we'll get to it via email. So the next question is for Jackie again, and it's quite related. So how does the TESSA program help drive down COGs other than in terms of yield?
All right, I'm sorry for the change in light.
It's all good.
So if we so for the decreasing the overall cost of goods, let's compare the triple plasmid to the Tesla program, right? So for the triple plasmid platform, especially in the upstream side, we see that we require a GMP grade plasmid to use for the actual transfection, as well as the associated transfection reagent, whichever that the client would prefer. But both of these, the plasmid itself and the transfection reagent, are very costly. And if you look at it, the larger the scale that you go up to, then the more and more of these starting reagents you would need for the actual production itself. So if you consider, then you have multiple of these large scale batches for AV generation, which would then compound the cost each time. What the Tesla Duo and the Tesla Pro system does is that because it's not using that plasmid and the associated transfection reagents, it's just an adenovirus co-infection, we can eliminate that from your process and therefore drive down the starting upstream cost as well. And the downstream process is, like I showed previously in the slides, it's the same regardless of whether it's triple plasmid or the Tesla Pro purification strategy, so that stays the same. So with the decreased cost from the upstream side, along with an increased yield for your starting and for your starting crude harvest all the way to your drug product. That way, overall, the Tesla Pro and the Tesla Duo program could give a very cost-efficient solution for any AAV-GR product you would like to explore.
Brilliant. Thank you. Thank you, Jackie. I'm sure our audience appreciates the answer. And speaking of cost of goods and cost in general, Dan, the next question is for you. So what are the key links of the chain to drive down the cost of gene therapy?
Yeah, I mean, I think everybody focuses on the manufacturing process, which Jackie's done an amazing job talking about how Tessa is increasing yields while also increasing quality. But I think a couple of the key things we think about as a therapeutic developer is dollar in, protein expression out. So at the end of the day, your gene therapy is being delivered with the goal of expressing a certain amount of protein in a certain amount of cells. And so multiple different levers that can be pulled to decrease that ratio. So, more effective amount of protein for a single dollar in. A couple of those levers that we've thought about pulling, and we have is, can you pick a GOI that's secreted and that can go to the liver? That immediately is going to lower your dose. Can you pick a more infective capsid that still produces highly? And so you're getting more bang for your buck on that side. Also more, promoter sets that are expressing more in the context of the cell type you want as well. And so all of these things I think need to be taken into account. I think the final part would be is also per your particular indication, how much expression do you need from these things? And so, multiple links to this chain, multiple levers to pull to try to decrease these, effective dollar in for protein expression out. And we're super excited to be working Advanced Therapies on really revolutionizing one of those links of the chains. But I think it's important to keep it in context of all those links in the chain to enable to create a scalable gene therapy.
Appreciate your answer, Dan. And the next one is actually for you as well. So what data is available on the long-term effectiveness and safety of AAV gene therapies in chronic disease patients?
Yeah, I mean, gene therapy is thought of as kind of a novel technology, but the truth is that we have had gene therapies in this context, AAV gene therapies in patients for over a decade. We've actually had it in dogs for over a decade. And so kind of, I'll take apart that a little bit. As far as expression data, people have seen consistent overexpression of AAVs in humans for over a decade, and they've actually similarly seen that in dogs as well. And so a lot of the preclinical models for the early gene therapies, particularly in hemophilia, utilized dogs. And so we have a huge amount of data that you're seeing that. So that lends yourself to believe that you would see consistent efficacy, considering you see expression for that time frame. In our hands, we've seen consistent overexpression of our gene therapies in dogs for approximately 3 years, which again is a long period of time for dogs, particularly animals where you're treating later in life. Going forward there, and we see consistent efficacy across that time frame as well. And so I think there's still work to be done to show this in humans and in multiple different chronic diseases. The short answer there is all the building blocks are there to suggest that you can get literally decades of efficacy from a single treatment. And as we're pointing out today, at COGS that are highly scalable to the point where you're able to treat, multiple different chronic diseases in animal health.
Fantastic. Thanks, Dan. And now we have time for a few more questions. So now our audience wants to know, are you using continuous ultracentrifugation? And can you elaborate on how you separate full and empty capsids? And what is your final full capsid ratio?
Right, so I believe that's a question for me. So for the UC portion, I cannot give you too many details into the actual parameters that we set. However, for the UC, it is a standard procedure of a certain, let's say, a gradient separation, in which that after the actual loading of the material into the UC tubes and the actual UC itself, we would then you typically would see a clear band between your full capsid and then your empty capsid. Then we would fractionate from those tubes and only collect what we would deem as the full capsid band. And from typically UC gives us a higher purity than, for example, other purification methods such as IEX. As I showed earlier in the slides, for UC, you could get up to 96% percent full of the capsid at the end. So it is, I guess, it's a more standardized procedure for the full and empty capsid separation. However, it is not as easily scalable the larger up you go. So then you could then use IEX chromatography instead for the separation itself.
Thank you, Jackie. So there's actually one more question. We're running out of time, but if you could answer that, that would be amazing. The audience wants to know: are you using AAVX for affinity purification?
So yeah, so again, I can't give too much details, but so earlier in the slides where I showed the typical downstream process platform, we do use a affinity resin that would only capture the AAV itself and basically have a flow through of all the rest of the impurities that are within the matrix of the harvested material.
All right. Thank you so much, Jackie. Actually, thank you both, Dan and Jackie, for answering those questions. And that's all we've got time for today. And any questions we didn't get to, we will reply by email. And the webinar will be available on demand tomorrow. So look out for an email from us with a link. And we would also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in resources on the right-hand side of the screen or in the on-demand email. Additionally, an attendance certificate is available on the right-hand side too. So all that's left is to thank Dan and Jackie once more for a great presentation, and thank you to our audience for listening. And we hope you'll join us again soon.
Thank you all.